Hello, everyone. My name is David Moeller. I'm the Chief Scientific Officer at a company called Poxel. And before we get underway, I really want to introduce myself. It's a great pleasure to be joining you today from Boston. I appreciate the invitation from Sarah Hunt and her colleagues at Alex TLC to share with you a little of our exciting information about two new development molecules that we have in clinical development for adrenomyeloneuropathy, or AMN, which is a subtype of adrenoleukodystrophy, a major unmet medical need. And before we get underway, I want to briefly introduce myself. I'm a physician by training and endocrinologist, which basically means that I focused my entire career on metabolic diseases. And I started at Harvard Medical School, focusing primarily on larger diseases like diabetes, and then moved into industry about 20 years ago, trying to develop new drugs that would help treat patients with metabolic diseases, including diabetes. But more recently, I've evolved and joined Poxel to focus on rare metabolic diseases, which include AMN and ALD. And I also have worked on other rare metabolic diseases, including lysosomal storage diseases. So it's a great pleasure to be joining you today and to share a bit of our story with you. So let me tell you a bit about our company before I talk about our strategy in particular. We're a small company based in Lyon, France, with groups of scientists and physicians, primarily in France, but also in Boston and in Tokyo. And our focus is to discover and develop new medications for serious, chronic, and rare diseases, and in particular those with an underlying metabolic pathophysiology. We are a clinical stage company. We have multiple molecules in clinical development, and we have a highly experienced management team that's been focused on clinical development as well as metabolic diseases. Here's a snapshot view of our clinical development and preclinical pipeline, different molecules that are in different stages of development. And we're very proud of the fact that our most recent, our latest stage molecule, now referred to as TWEMIG, has been actually approved and launched as a new therapy for type 2 diabetes in Japan. So that just underscores that we have a lot of experience in drug development, taking molecules all the way from very early stages through to becoming approved therapeutic agents that are now being marketed in various geographies around the world. We have two mid-stage molecules that we are developing for two different approaches, for two different disease indications. These two molecules we call PXL065 and PXL770. And as you can see, they're both being developed for a disease we call NASH, which stands for non-alcoholic steatohepatitis, or in other words, fatty liver disease. We're not going to talk about that today. What we are going to talk about is how we've been able to reposition these molecules and develop some new information that really provides the basis for their approach and being developed now for patients that have adrenoleukodystrophy, the adrenomyeloneuropathy form, as well as potentially childhood cerebral adrenoleukodystrophy. And these two molecules have different properties, as I'll talk about later on, in terms of how they work. But they're both basically molecules that are in fairly advanced stages of clinical development. As you can see, for liver disease, these molecules are actually have been and are in fairly large clinical trials that are ongoing at the moment. But we also have the opportunity now to advance these molecules directly into patients with adrenoleukodystrophy, which is what I want to focus on today. To give you a bit more of a perspective about the company and our strategy, we started as a company that was really focused more on diabetes, a classic metabolic disease, evolved into focusing on fatty liver disease, or NASH, and have only within the past year or so begun to really focus on leukodystrophies, and in particular on X-linked adrenoleukodystrophy, or ALD. And as I mentioned, we have two molecules now in clinical stage development for this disease area. So why did a company that was focused on type 2 diabetes decide to move into rare metabolic diseases? And I just want to give you a bit more of an understanding about that. And first of all, relative to diseases like diabetes, for example, patients with rare metabolic diseases have extremely high unmet medical need. There are very, very few therapies available for any of these diseases. And as many of you know, rare diseases in general are quite numerous, and perhaps as many as 1 in 10 people have some form of a rare disease. And importantly, for a small company like ours with only 55 employees, we really have an ability to have a major impact by targeting a rare metabolic disease rather than going after something much larger like diabetes. In addition, for a company like us, Poxel, we have a lot of experience in metabolic diseases, I mentioned, and it's clear that there are a large number of rare metabolic diseases, over 1,000, and there's can be divided into these categories shown on the lower right here, where disorders related to fatty acid metabolism in particular are areas of interest to us and are areas where the diseases like adrenoleukodystrophy, like adrenomyeloneuropathy, and other leukodystrophies, including Zellweger spectrum disorder, for example, could be categorized. And we have important molecules in these areas, as well as proven abilities to develop new molecules for new indications, as I've mentioned. Over the past year, we've been working a lot with relevant patient advocacy groups, such as Alex TLC and others, and also with a number of real experts in the field of leukodystrophies. And we're now very excited to be advancing our programs. So what is our view of adrenoleukodystrophy and adrenomyeloneuropathy? One thing I want to emphasize, and this is a little bit technical, is that these diseases, adrenoleukodystrophy and its large subtype adrenomyeloneuropathy, are all due to specific mutations in one particular gene called ABCD1, which I'm sure many of you are familiar with. And the consequence of mutations in this gene are that you have very elevated levels of long-chain fatty acids, so-called VLCFA, or very long-chain fatty acids. The gene involved is called ABCD1, which we don't need to get too technical, but it involves the ability of cells to transport and then metabolize these very long-chain fatty acids. These elevations in very long-chain fatty acids cause damage to multiple organ systems, in particular the adrenal gland, resulting in Addison's disease, as well as in the neurological system and the nervous system. And we think that our two approaches, which we call D-TZDs, and I'll talk a bit more about this later, and AMP kinase activators, I'll define those terms in a few minutes, are really well suited to targeting the disorders associated with adrenoleukodystrophy. Our view of adrenoleukodystrophy, which I assume is shared by many of you, is that it is really not such a rare orphan metabolic disease. It is a neurometabolic disease involving both the metabolic system as well as the neurologic system as a secondary consequence of the metabolic derangements. And it's fairly prevalent as rare diseases go. In the US, we're estimating now about 20 to 29,000 patients. And this is largely because of the increased recognition of more and more patients being diagnosed through newborn screening, which is now fairly commonplace in the US, as well as becoming available in parts of Europe, and we hope in the UK fairly soon. I think, as many of you know as well, adrenoleukodystrophy has two major subtypes: the cerebral form of the disease, as well as the adrenomyeloneuropathy form of the disease, or AMN, which is sometimes referred to as spinal cord disease. And the way that we look at this is that, of course, children are frequently affected with cerebral disease, and there's a peak in the incidence around age 10, and this is perhaps the most devastating form of the disease in that very severe brain lesions can develop. But then during adulthood, beginning in late adolescence and throughout life, there's a progression of symptoms associated with the spinal cord disease, or AMN, in almost all, or essentially all men, because this is an X-linked or X-linked disease, which afflicts men more so than women. But the symptoms of AMN are also fairly common in women, and it's estimated now that about 80% of women will ultimately develop perhaps milder, but still very, very significant symptoms associated with AMN. And another important point for us to keep in mind in developing drugs in these areas is that cerebral disease not only develops in childhood, but can occur at any point later on in adulthood as well. So many patients with AMN, in particular men, can develop devastating cerebral disease at any point during the course of their disease associated with the spinal cord. Now, the AMN form of the disease can be mild, as is the case with our friend Ben Lanile here, who's one of the co-founders of ALD Connect. But he does have significant impairment, and he feels quite strongly that we really do need therapies for fully adult men who are in the prime of their lives and have deep deficits in terms of gait, in terms of balance, in terms of bladder function, sexual function, etc. So we feel, along with you, that there's a really compelling need to develop new therapies, and our initial focus will be on adults with AMN, but we may also be able to prevent or delay the occurrence of cerebral disease in those adults, and potentially then to treat in the future children who are afflicted with cerebral disease. As you also know, there are really no approved pharmaceutical therapies specifically for AMN or ALD. And the only real therapy that's been proven to work successfully is hematopoietic stem cell transplant, or HSCT, where there is good evidence now that there are fairly dramatic improvements in longevity and in the ability to arrest the cerebral disease, providing that the intervention occurs very, very early in the disease process. And it really can only be targeted to children, say, under the age of 12 to 14. So as I said earlier, there are a large number of adult men who may develop cerebral disease for which there are no effective options whatsoever. They are not candidates for hematopoietic stem cell transplant, and they would be candidates for an additional intervention, such as the molecules that we're developing. And in addition, there are a fairly significant number of children for whom hematopoietic stem cell transplant is either not appropriate because they've been diagnosed too late, or they may not be fully responsive, and there could be interventions that can be targeted to that population as well. We also feel strongly, having consulted with a number of the experts in the field, that the field is really evolving quickly, such that a number of different tests can now be employed to effectively diagnose the disease, to track its progression, and more importantly for us, to be able to monitor whether our drugs are actually working, in particular in early stages of development, before we may have the opportunity to fully evaluate the symptoms in a longer-term context. So let me tell you a bit about our new therapeutic candidates for AMN and ALD. And to start off with, this is again a bit technical, but I'll try to explain it. We have two completely independent approaches that result in significant benefits, we think, potentially, for adrenoleukodystrophy. On the left, you'll see that we have a set of molecules that are involving the ability to target a specific protein that's present in all cells and tissues in the body called AMP kinase. AMP kinase is a very important kind of energy regulator or energy sensor that exists in many cells and tissues. And in general, it has the ability to regulate inflammation, which is a key component of the disease for ALD and AMN, and also to regulate metabolic pathways such as lipid or fatty acid metabolism, which again is also a critical component of the disease process for ALD and AMN. On the right, we have a different approach, which involves a series of molecules that we call thiazolidine diones. Never mind the specific acronym here, but these are molecules that have a specific chemical structure. And by modifying them in a specific way, we can segregate one of two major pathways or major effects of these molecules in order to emphasize what we think are the preferred approach, effects of these molecules at the expense of a reduced effect on what we call the genomic pathways, which have been associated for these types of molecules with weight gain and edema, fluid retention, etc. With these molecules that have the modification that I mentioned, we can accentuate the pathways or the biochemical effects that we think are potentially beneficial, especially for adrenoleukodystrophy. And those would involve pathways and approaches that can adjust cell signaling, also cause anti-inflammatory effects, and enhance lipid metabolism that could be beneficial in terms of lowering the very long-chain fatty acid levels. So I won't belabor this, but it's a complicated system. I'm coming back now to the AMP kinase story, where this has been traditionally viewed as an approach to major metabolic diseases like diabetes and fatty liver disease. And as I mentioned, has a host of different effects that could be beneficial for not just these sort of diseases, but also for rare metabolic diseases. And in the case of the other class of molecules, where PXL065 is our lead molecule, as I mentioned, we now are accentuating these particular effects of these molecules, which in turn are associated with an anti-inflammatory effect, an ability to accelerate lipid metabolism that could be important, and also can be cell protective in the case of situations where cell death is a potential effect. So what are we actually talking about? We're talking about two different classes of molecules, and for each of those two classes of molecules, we have one advanced lead molecule. So PXL770 is the most advanced lead molecule in this category, the AMP kinase activator class, and PXL065 is the most advanced molecule in this category, which is the modified thiazolidinedione molecules. And this particular molecule is also a derivative of pioglitazone, which, as I'll show you, is a common drug that's been used for type 2 diabetes in the past. What's important to keep in mind about both of these molecules is that they have been studied extensively in preclinical toxicology systems where we're looking at the effects of high doses and ensuring that the molecules are safe in animals. And then subsequently in multiple clinical trials that have been conducted with each molecule, both in healthy subjects to evaluate their overall safety and whether we have adequate levels of the drug in the bloodstream, and also in specific studies that are ongoing or have recently been completed, looking at other diseases such as fatty liver disease, where, as I mentioned, both of these molecules also have some strong evidence to support their potential utility. So now I want to just give you a glimpse, and again, this is very technical, but I'll try to explain it at a high level, of why we're so excited about our particular molecules. And it's based largely on a series of experiments that were done in the laboratory with external collaborators and experts in the field. And in the case of both of our lead molecules, we have really interesting data that strongly implicates these molecules as having real potential for adrenoleukodystrophy. So starting with the AMP kinase activator, this is the PXL770 molecule I've been referring to. There are some published scientific literature that do support this hypothesis. And then based on that, we took our lead molecule, PXL770, and we put it through its paces in a number of experiments. Those experiments involved using human cells that came from patients, directly from patients who have AMN and ALD, as well as using a classical animal model of the disease, which is a mouse that has basically recapitulated the phenotypes associated with disease. And over here on the left side in these two panels, I won't go into detail, but suffice it to say that 770 has a really interesting ability to normalize or largely normalize these elevated levels of long-chain fatty acids that exist in the cells that come directly from patients. And this could in part be due to a restorative effect to increase the expression or the presence of another transporter that's not mutated in the case of the disease, but could serve as a way to rescue the cells. And then over on the right, you can see that these effects that we saw initially in patient cells can also be recapitulated in the animal model of the disease. And in particular, we see a significant decrease in the spinal cord, which is a very relevant cell type and tissue type for the disease of the elevated long-chain fatty acid levels. And we have a pretty similar profile with the other series of molecules, the TZD series, which is exemplified by PXL065, our most advanced candidate there. And here again, there is a literature that implicates these molecules or these kinds of this kind of molecule in the disease, and that comes from studies that were done with pioglitazone, which is a marketed drug for diabetes, where it was able to reproduce some of the effects that I showed you earlier in the animal model of disease by partially normalizing the disease. So based on this and based on our understanding of the mechanisms underlying how our molecules are working, we again took our lead molecule, 065 in this case, and studied it in very similar systems as I just described you for the other lead molecule, in the patient-derived cells, which are the two left panels here, and then again also in the disease model, the animal model, which is the mouse model of disease called the ABCD null mouse. So here on the left, you can see that, again, the VLCFA levels, the long-chain fatty acid levels in the cells from the patients are markedly elevated compared to normal. And we can essentially normalize those levels by exposing these cells to our molecule. And again, it may be in part due to the rescue effect of this other transporter that's not mutated and being turned on and increased in its levels. In the animal model, we can also see that in particular with our molecule, which is the green bar here, we can largely reduce the elevated levels of these toxic long-chain fatty acids in the spinal cord. And that's even better than you can see with the related molecule called pioglitazone that was the subject of some of this earlier literature. Some more recent data is shown on this slide, and this is just examples of some of the data that we've also derived from the animal model that I referred to, the disease model, which is the ABCD1 mouse. And here you can see that we're looking at under the microscope, an electron microscope in particular, at the phenotype, at the disease, directly involving the nerves in the body, in the sciatic nerve in particular, which is a large nerve, comes from the spinal cord. And you can see that normal mice have these fairly symmetrical, round-shaped neural cells, and they become distorted in their shape in the context of the disease. And then in response to treatment with our molecule, in this case 065, we can really largely improve the situation in terms of what's visible under the microscope. And that's shown on the right here, but also quantitated in this graph below. These effects that you see here were very, very similar to what we also observed with our other molecule 770, which is not shown on this slide. And then in addition to these effects that you can see under the microscope, we also looked at behavioral tests that can assess the ability of these mice to function in terms of a potential neurologic deficit. They don't have extremely severe neurologic deficits like some patients do, but they do have a subtle phenotype, a subtle effect, where one of the effects that they demonstrate is a difficulty in crossing a beam. It's called the balance beam test. And you can see that that difficulty is scored, and it's worse with the disease model. And then we can basically restore that situation towards normal with chronic treatment with, in fact, both of our molecules, but the data that's shown here is just illustrating the effects of 770. So what I've showed you very quickly is that for both of our two advanced lead molecules with different underlying mechanisms, we have a fairly robust set of data involving effects on cells that come from humans with the disease, with ALD and with AMN. And these are associated with basically improving the function of those cells and very remarkably lowering the toxic levels of elevated long-chain fatty acids. And then in the animal model, we have a variety of beneficial effects, both at the level of what we call biomarker signatures, like reducing the long-chain fatty acids in various tissues and plasma, improving the images under the microscope of the nervous system, improving the neurologic behavior, as I showed you on the previous slide. And for both of these molecules, we have fairly large data sets right now of clinical safety in other disease states as well as in healthy subjects. And when we compare our data to several other molecules that are in development for the same disease, all of these are targeting adrenomyeloneuropathy initially, as we are. We think we have a very favorable profile overall, and relative to some of the molecules, more advanced molecules that could be exploited in early clinical development. So what is our approach, and exactly what are we going to be doing over the next few months? We're very excited to have recently announced that we are going to be advancing in the early part of 2022 into two separate clinical trials. We call these phase 2A studies in patients with adrenomyeloneuropathy. So these are going to be adult male patients with the spinal cord disease, AMN, age 18 to 65, with no active cerebral disease, and we will study 12 patients in each of these 2 trials. So we're basically conducting 2 parallel phase 2A trials for a total treatment duration of 12 weeks or 3 months. And we will be studying at least 1, perhaps 2 dose levels in each of these 2 trials. So these are mirror image studies, one designed to focus on 770, an identical trial designed to focus on 065. And what we're going to be measuring in these clinical trials are primarily what we would call short-term benefits, potential benefits related to the disease. In particular, we're going to be looking for potential reductions in the very long-chain fatty acid levels, which are clearly elevated in the blood, and you can measure that readily. We'll also be looking at another important potential marker of neurologic damage, which is called neurofilament light chain, as well as additional exploratory biomarkers. So we don't believe that this 12-week duration will be enough time to necessarily see a clear improvement in symptoms associated with the disease, but it will be enough time, we believe, to evaluate the potential in the disease and to be able to allow us to select which of these 2 molecules we would prefer to really move into advanced stages of development. And that's the goal, is to choose one of these 2 molecules following the completion of this study towards the end of next year, and then to begin a potential pivotal trial in 2023. So what does that overall scheme look like? It's depicted on this slide. As I mentioned, we're going to be conducting the 2 smaller studies in 2022, completing those studies by the end of 2022, and then sometime by the end of 2023 to be able to launch a longer term or 1 or more longer term pivotal trials, i.e. a phase 2B or phase 3 study, having chosen one of these 2 molecules as the best candidate to advance towards approval and potentially commercial launch within the next several years. So to summarize, I'm very excited that we have the opportunity to work with organizations like ALEX TLC, and we are also working closely with several other key organizations around the world that are really helping us understand how we can best impact the disease, understanding what really matters to patients. And for us as a small biotech company, we believe that we have an opportunity, along with other companies, and potentially to combine our therapies in the future, where we can have a really big impact on a very, very important disease, adrenoleukodystrophy and adrenomyeloneuropathy. We believe that we also have good clinical safety profiles for both of our 2 advanced lead molecules, and that we have very interesting and we think compelling data based on the preclinical models that I shared with you from the patient cells as well as from the animal model, with the potential to accelerate the approval, potentially, based on biomarkers and early signs of benefits. And we would like to continue to stay very closely engaged with ALEX TLC and other organizations, as well as to hear more directly from patients about how we can better optimize our development programs. Thank you very much for your attention, and I really appreciate the opportunity to share with you.